Okay, so proteostasis is the synthesis, assembly, and degradation of proteins in vivo. Okay, so synthesis of proteins, that's going to take place at the ribosome. Now, that protein that comes off of the ribosome is going to fold. It's going to be a folded intermediate. So in the process of going from that polypeptide produced from the ribosome to the functional protein, it's either going to fold by itself or it's going to use the help of a chaperone. Now, that is what I'm talking about right here, going from here to here. If you do that, consider that to be a good thing. Okay, that native folded protein can go on to work and do its job, and then when the time comes, it gets broken down into individual peptide fragments. Those peptide fragments can lead to the, well, basically turnover of amino acids so you can synthesize more proteins. Okay, now let's take this, so that's kind of path one. Path two is your folding intermediate doesn't actually go to the proper place. It doesn't actually fold properly. So what might happen is your protein gets misfolded. Okay. Now, when your protein gets misfolded, it has basically two fates. One fate is that it can be targeted by the proteasome and broken down into individual peptide fragments, or that misfolded protein can aggregate together to form amyloid fibrils, oligomers, or amorphous aggregates, which can potentially be dealt with by autophagy, and you result in these peptide fragments. Those amyloid fibrils could ultimately be deadly to the cell. Now, our third path is that folding intermediate could go directly to disaggregation and formation of these amorphous aggregates, an amyloid fibril, and then ultimately to these peptide fragments. So maintenance of protein activity is accomplished through all of these different ways. The ideal direction is your protein gets folded properly, be it with the help of a chaperone, and that chaperone is just like the term when you talk about a junior high school dance, there's something that helps it along. That protein helps your protein fold properly. We'll get into that a little bit later in just a few slides, but you've got a lot of things going on here. You've got the production of the properly folded protein, breakdown of that properly folded protein. Now, one thing that if you go on to take metabolism, you will talk about is taking this misfolded protein right here to your peptide fragments using the ubiquitin proteasome system. That ubiquitin proteasome system is a pretty cool system where ubiquitin is a small protein that is added to your protein, and that's what targets it to the proteasome for subsequent degradation. Protein stability and folding. A protein's function depends on its 3D structure. Now, loss of structural integrity and accompanying loss of activity is called denaturation. So a protein can be denatured or rendered ineffective, or not ineffective, but rendered dead basically through a couple of different ways. It could be rendered dead through physical, solution, the environment. It could be too hot or too cold. It could be exposed to an extreme pH. Organic solvents or chaotropic agents, which like urea or guanidinium hydrochloride, which are going to disrupt hydrogen bonds. So basically, it causes chaos by disrupting one of the bonds that you rely on for stabilizing your molecule or your protein. Okay, so this right here is a protein denaturation curve. What we've got is ribonuclease A, and this is the y-axis is percent unfolded. So this is going to be unfolded until you reach about 55 degrees Celsius. And as you go up, once you hit about 80 degrees Celsius, ribonuclease A is sensitive to the temperature to the point that it is completely misfolded at about 80 degrees Celsius. Ribonuclease A, in graph B, I'm going to label it, is subjected to guanidinium hydrochloride, and a concentration that is a substance that breaks up hydrogen bonds. At up to 2 molar guanidinium hydrochloride, you're going to be fine. It's not going to disrupt enough hydrogen bonds to render the protein fully misfolded. But then once you get closer to 5 molar guanidinium hydrochloride, that protein has completely been misfolded. Okay, ribonuclease A, or ribonuclease refolding experiment is really a seminal study in protein folding, and it was undertaken by a guy named Christian Anfinsen. He was awarded the 1972 Chemistry Nobel Prize for it. And essentially what he did was he studied the structure of the protein ribonuclease A. It's a small protein. It contains 8 cysteines that are linked via 4 disulfide bonds. What he did was he added 2-mercaptoethanol, or beta-mercaptoethanol, and urea. And what he wanted to do was denature the protein. So urea is acting as a chaotropic agent. Beta-mercaptoethanol, or 2-mercaptoethanol, served to break up disulfide bonds. So what he did was he added those 2 agents. He broke up the disulfide bonds and the hydrogen bonds, and that protein, well, it was unfolded. It was unfolded and denatured. Then he titrated away the urea and the beta-mercaptoethanol, and lo and behold, the protein spontaneously refolds. So the correct disulfide bonds were also reformed. And basically what the takeaway from that was the primary sequence, so your amino acid sequence, AA sequence, was enough to determine the native conformation of the protein. Now here's a very nice schematic of someone's almost life work, basically. You have your native catalytically active protein, you added urea and beta-mercaptoethanol, and you produce this unfolded state. Disulfide bonds are all broken, you have these individual cysteine residues floating around, no disulfide bonds formed. Then you dilute it, and you remove the urea and the beta-mercaptoethanol, and lo and behold, disulfide bonds are reformed. That protein reforms. That protein is now functional again. So Christian Anfinsen's work was very valuable, and ultimately it kind of gave way to structural biology and getting to a point where you understand how proteins fold. So proteins folding follow distinct paths. So here's a 56 amino acid peptide. And when you look at just different parts of this protein, so one of the ways that this protein is going to fold is by first amino acids 8 to 15 folding. 8 to 15 are going to form a shape, then 6 to 17 are going to form a shape, then 1 to 20 are going to form a shape, and that's going to fall into this 1 to 56 overall structure. Now, if you looked at and you isolated and made a polypeptide of only amino acids 30 to 37, they're going to take on a shape. Then if you look at 28 to 39, that's going to take on a new shape that encompasses the same 30 to 37 structures. So then 45 to 52. So basically what this was a series of experiments. Somebody got amino acids 8 to 15, and they looked at the structure that those formed. Then they looked at 30 to 37. They looked at the structure that those formed, in 45 to 52. And then they kept expanding. And so you have in the second phase almost, the second stage, you got amino acids 6 to 17. What did those fold like? Oh, well, they kind of folded in a similar way to 8 to 15, but a little bit different. Well, it makes sense that they had part of that because, well, they had, I guess, more to work with. Now, from there, you looked at amino acids 1 to 20. And so basically this gives a process and kind of a hierarchy on folding of proteins. So how can proteins fold so fast? Proteins fold to find the lowest energy fold in microsecond to second timescales. So how can that happen so quickly? It's mathematically impossible for protein folding to occur by randomly trying every single conformation until the lowest energy one is found. So there's a search for the minimum is not random because the direction toward a native structure is thermodynamically most favorable. So this second part here, I remember hearing it characterized if a protein went through every single different permutation in which it could fold, it would take longer than I'm not exactly sure on the time interval there, but it would take longer than the life of the known universe to actually form that lowest possible scale, or lowest possible fold. So the free energy funnel model of protein folding is dependent on stability of motifs within the protein. Okay, so these different models, think about this kind of perimeter of this circle here as starting points for a unfolded protein, and then N is going to be your native fold. Okay, so it's entirely possible that what happens is basically it just begins to find a lower energy model until it finds the lowest. Okay, so it's just going to consistently be folding into a lower model. So multiple pathways lead to one stable outcome. Another idea is that multiple stable intermediates lead to a final fold. So we might start at energy level, go lower, and then go a little bit higher, and then go lower, and then form and get to our native level. Now, another proposed idea is that the immediate folding of one stable outcome, so these basically just exist as an unfolded form until, boom, they plummet into the properly folded lowest energy form. Significantly stable intermediates are overcome before a final fold, so it's beginning in a lowering energy, and then it has to go to a higher energy fold, and then it goes to the lowest possible energy fold. So chaperones are something that I mentioned previously, and they're assistants in folding folded intermediate forms of proteins. So the way that chaperones work, and this is just an example of a chaperone. So what we have, let's start in the middle with our folding intermediate. That folding intermediate could potentially go to a native protein, and boom, that's where we want to be. Alternatively, that folding intermediate may not get to that folded state, in which case it's going to aggregate with a bunch of other folding intermediate proteins. Alternatively, in the direction that I want, so it could also fold into this kind of just amorphous protein that doesn't necessarily have any function. It's also not going to aggregate with anything. It's just kind of, it's there. That and the folding intermediate are both going to go to this protein right here, HSP70. This is a heat shock protein, HSP70-ATP. So what that means is this is a protein that is bound to ATP, and what it's going to do is it's going to receive these fully folded, although not particularly functional, or folding intermediate proteins. And what it's going to do is it's going to refold this protein. And the way that it's going to refold it is, it's going to refold it, and the energy that it's going to use to do that is by the hydrolysis of ATP, which is why this PI comes off of it. Now, what that's going to do is it's going to refold this protein, and it's going to release it ultimately. It's going to release that folded protein. Then it's going to be the HSP70, which is bound to ADP, and it's going to replace that ADP to ATP. Now, one model that I think is a better illustration of all of this is known as the GROEL GROES complex. Now, for this, what we're going to do is we're going to start in the middle. Okay. Now, when we start in the middle, we have our native protein, that's our destination, our native protein or our slow folding intermediate. Now, that's where we want to be. But let's go back to this point right here, our folding intermediate delivered by the heat shock protein. Now, GROEL and GROES, these are two different parts of this protein. So GROES is this little yellow cap. Okay, it's this little yellow cap there and throughout this whole system. So when a protein is, when GROEL GROES complex is ready to fold a protein, GROEL is separate from GROES. Okay, GROEL will bind to that folding intermediate. It will also bind to 7-ATP, and then it will bind to the GROES complex. It's going to hydrolyze those 7-ATP molecules to make 7-ADP. Now, those 7-ADP molecules, sorry, I got mixed up there. When our 7-ATP molecules, our protein and GROES have basically come in contact with one another, what we're going to do is we're going to hydrolyze that ATP, releasing 7 inorganic phosphate molecules. So now we've got 7 ADP that are associated with GRO-EL and GRO-ES, and our protein is slowly but surely being refolded. What's going to happen then is we're keeping track of only the top half of this. So the top half of that, we've got ADP now, our protein is being refolded, and then all of a sudden, our GRO-ES complex is, or our GRO-ES is released, and our protein is released into the center, or into the center of this model. So now we have a slow folding or a natively folded version of our protein. What's important about this, and what's kind of complicated about this figure, is that the GRO-EL GRO-ES complex basically has 2 parts that can fold proteins. So we are just tracking the top. The bottom half of that simultaneously is receiving a folded intermediate, it's binding ATP, it's going to bind the GRO-ES complex, it's going to hydrolyze our ATP, it's going to refold our protein, it's going to release GRO-ES, it's then going to release our natively folded protein. So the point of this all is, when you look at this figure, keep track of what is binding to what and what is happening in the bottom half of that. So think about this as just as, let's take this part right here, this step right here. As one GRO-EL has bound ATP and our folding intermediate protein and is beginning to bind GRO-ES, the other part of the protein, the other GRO-EL subunit, is releasing GRO-ES. It's also releasing its natively folded protein. So just as one protein has been refolded, another protein's folding has completed. So that's being released into the system. Now here is a nice model of what the GRO-EL GRO-ES complex looks like. This shows us with our 2 GRO-EL kind of cylinders right here. GRO-EL, GRO-ES. And here is our newly folded protein, GP23, and here is a non-native folded GP23. So what's happening in this is, you can see almost, and I don't know if this was intended or not, but our GRO-ES here is about to be released, and then another GRO-ES cap is going to come on here, because we've got our newly folded protein and we've got a non-native folded protein. So protein misfolding is actually not a disorder, but it is implicated in a number of different diseases. For instance, mad cow disease, I believe it's Crutchfield-Jakob disorder, Huntington's disease, and Alzheimer's, the A-beta peptide, or beta amyloid. So misfolding of proteins is kind of no joke. Native beta amyloid is a soluble globular protein. Misfolded beta amyloid promotes aggregation at newly exposed protein-protein interfaces. And the corrected folded helices are lost, and peptides from beta strands, beta helices, and beta sheets are ultimately what kind of result from that. Well, I believe that's the end of this chapter. Let me make sure of that. This is chapter number 4, 3-dimensional proteins. Yeah, that would be the end of this chapter. So this is kind of a lengthy one. Next one is going to be function of proteins. We're going to hone in on oxygen binding proteins, but I hope this is helpful, and have a good one.